was correct. I think I have read it's 50 to 85% of the oxygen in the atmosphere is produced by algae. Some of the 50% quotes are just phytoplankton in the ocean, but then if you take all of the bays, wetlands, inland water bodies, I think it gets higher. Just a quick outline of today's talk is to give you that it's overall 4 parts, a kind of introduction, then a little bit about the Maryland Phytoplankton Monitoring Program. I'll talk about marine harmful algae and then freshwater harmful algae, and a sort of kind of pulling it together that is based on Pat Gilbert's work at University at Horn Point. Before I get too far, I always like to mention that we have not had any human illnesses or shellfish closures due to algae bloom. So in case I forget to say that later when I start talking stuff and it sounds really scary, just know that in the Maryland and Chesapeake Bay region, we have been very lucky where most other parts of the US have had these. And so we monitor to make sure that we do protect human health and that we figure it out, but knock on wood. It's not advancing again. Okay. So as we just talked about, algae are one of the primary producers in the Chesapeake Bay. They help, like all plants, produce oxygen. So most people, when they think about the Chesapeake Bay, we talk mostly with the Bay program about submerged aquatic vegetation or seagrass beds as important habitat and primary producers in the Chesapeake, but there are also algae, both macroscopic and microscopic. So macroscopic includes seaweeds and things that most people don't like, but are part of the ecosystem. And then mostly what I'll be talking about today are the microscopic algae or phytoplankton. So one thing I like to talk about in terms of the primary producers in the Chesapeake Bay is that as you change nutrient loading within the bay, you do change your primary producers. So in optimal seagrass habitat, you have low nutrients, but not zero because plants need nutrients. As you increase nutrients, you get more light limitation in the water, and that allows for more macroalgae or epiphytes to grow, either both. And then as you increase nutrients even further, you get much more algae in the water. The phytoplankton made up of all different types of species. One thing we do to measure phytoplankton abundance is measure chlorophyll A in the bay, but today I'm going to be talking about the actual species and different things. So obviously, I think all of you will know algae, like a lot of things, are very diverse. There are over 1,500 species within the Chesapeake Bay with all sorts of different sizes and shapes and colors. They do produce oxygen and are the base of our food chain that help feed all of our fish and shellfish. As I mentioned just before, as you increase nutrients, you increase the biomass of these algae in the water column. And we have about 30 of these species out of the 1,500, so less than 1%, do have the potential to produce toxins or have negative effects such as fish kills. So I just wanted to do a little bit of an introduction. Most of you probably know all of this, so I won't spend a lot of time on it. But what do algae blooms consist of? They are an overgrowth of algae, right? So some algae is always natural and needed. It helps feed the ecosystem. But when you have an overgrowth of this, you get pictures like this where you get water discoloration, you get a lack of clarity to the seagrasses. It can smother shellfish underneath when it robs oxygen from the water column when the biomass decays. There's smell and odors associated with these things. And that's part of why we care about them. I just want to kind of point out there's 2 types of algae blooms. You have the ones in the water column, and that's what we normally routinely monitor for when we measure chlorophyll A and other things. But there's also benthic algae that grow on the bottom and sometimes late in the season come up to the surface. And this is something that we do not have a routine monitoring program for, but over the last few years, we've shown that we need to expand our monitoring, and I'll get more into those later. So again, these algae blooms are widespread throughout Maryland. They are diverse. I tend to not always like color, like red tide or black tide, or maybe you've heard recently pistachio tide in the news a lot. That's actually an eruption of sulfur bacteria, and not even, it's when we have an inversion in the anoxic water comes up, but it turns it kind of a creamy green color. So there is quite a diversity of how the blooms look in the bay, but then there's even more diversity when you get to looking at them under the microscope. So there are the algae blooms that might cause ecological impact, and then there are nuisance blooms, blooms that may not look attractive, may smell bad, may impact jet skis or boat propellers or impact fishing, things like that. As I mentioned, there's over 30 species that are toxigenic in Chesapeake Bay waters. This is just a few of the ones that we see to kind of show the diversity of shapes. And so to get more into our phytoplankton monitoring program in Maryland, most of what we do is to analyze routine phytoplankton monitoring. That's fixed monitoring at a set number of sites that are monitored once to twice a month. We do routine phytoplankton, but we also do a subset of those sites for picoplankton. So those algae that are less than 5 micron and difficult to assess with routine microscopy need a specialized technique to look at them. But overall, those two types of monitoring we use to kind of analyze the status and trends, any changes in the community. Then the next level of phytoplankton monitoring that we do is HAB response, which is short for harmful algal bloom. So we do this when we get reports in either our website or an 800 number that we have in the state, or our field crews are out monitoring, happen to notice discolored water or something that's unusual. People that live on the water give us calls all the time. And so we'll go out to check those blooms to make sure that things are okay, that there's no potentially toxic species to protect human and animal health. We also work a lot with university partners to do special studies to increase our understanding of bloom formation and processes. We were recently doing a special study in the Susquehanna Flats because there's been a lot of a benthic algal cyanobacteria called Lingbia, or now Microceria woolii, up there, and we were trying to see if those things were impacting the seagrass growth. And then at the very top of our little pyramid is testing for toxins. And so the toxins can lead to no contact advisories with the water or fishery closures. And so often we send these, our sister agency, Maryland Department of the Environment, can test for many of these toxins, or we send them out of house to expert labs. We do have some rapid testing that's also conducted to quickly make determinations. This is a map of all of the water quality monitoring stations that we do for standard parameters such as nutrients, oxygen, temperature, and chlorophyll. At a subset of these stations, we also take samples for phytoplankton analyses. As you can imagine, sitting at a microscope can take 45 minutes to an hour to do a full community assessment of that phytoplankton. So this is a map. The black dots are the substations that we analyze for phytoplankton monthly in a given year. But the red dots are all of those harmful algal bloom response monitoring events that we might have had during that year. So you can see the distribution of the routine sites as well as those event sites. So our routine data historically was used to put into a multi-metric index called the Phytoplankton Index of Biological Integrity that you're probably familiar with other IBIs. It gives you an average score for both summer and spring of the phytoplankton condition. And this lets managers kind of know if the phytoplankton is good food quality or other issues for that particular area. And at the bottom there is the paper that this was based on by Richard Lakacher and Claire Buchanan. Richard was at Morgan State and Claire is at ICPRB. So I thought today I'd also discuss a little bit of these kind of new or additional tools that we've been using to help assess phytoplankton communities. The two with the stars I'm going to talk about in a little more detail, but I thought I'd put them all up here. So we have started about 10 years ago working with NOAA to help use satellites to detect areas of high chlorophyll. And then we've also been working with them to develop algorithms to hopefully narrow down to the phytoplankton group or class what it is. There's also a NASA app that's called Hydrocolor, and citizen monitors use this, but we also use this when we go out. You download it and you take 3 pictures, one of this black card that's or gray card that's used by photographers that help calibrate your phone camera, and then you take a picture of the water and you take a picture of the sky, and it is able to send NASA information on the red, green, blue bands that are being sent back, and that helps them ground truth the satellite data, and hopefully eventually will help us detect blooms with just those kind of tools of your camera, your phone camera, and a photographer gray card. We've tried this small handheld iolite field scope to detect blooms. It has this Bloom Optics AI technology app. It's for use in the field to help estimate cyanobacteria and cell densities. So the image on the bottom right is a microscopy image from the iolite field scope, and then overlaid all the boxes is that AI technology recognizing different species and putting them in boxes, and then it comes up with a cell count. We do find this works pretty good in a significant bloom like this one, but sometimes when concentrations are lower, it has not been as accurate. We also use continuous monitoring units that are deployed around the state that give us indicators of chlorophyll, dissolved oxygen, pH, and also sometimes phycocyanin, which is indicative of cyanobacteria. These are monitored every 15 minutes and sent to a website. I did put our Eyes on the Bay website in the group chat, so later you can take a look at that for both phytoplankton, harmful algae bloom data, as well as water quality if you're interested. I also talk a little bit more about some new imaging technologies we are using in conjunction with University of Maryland and St. Mary's College to help rapidly detect potentially harmful algae. We started instituting with NOAA a volunteer phytoplankton monitoring network. We have put that out in the coastal bays, as well as we have instituted a aquaculture phytoplankton monitoring network. Currently, I think we have 5 aquaculture groups working with NOAA and us to do that. We also put out passive toxin sampling devices. They are unfortunately called SPAT, which is not to be confused with oyster spat, but they can be put out for 1 to 2 weeks, and they passively absorb toxins in an environment to let us know if there has been potentially toxic algae in that area. We are working with the Virginia Institute of Marine Sciences to develop models and forecasts for potentially harmful algae. So now I'll talk a little bit more about the satellite and imaging technologies that we're working on. It will advance. There we go. So this is an overview of how we use satellite to help us detect blooms. On the top left is just a true color image of the Chesapeake Bay region. The second one, that's called relative band is red band difference, is one algorithm that we use to help determine if there's a dinoflagellate bloom, so one particular group of phytoplankton. And then all the way on the top right is just overall chlorophyll A, so that we know there's an accumulation of algae biomass. Then if you look at the bottom, they're the zoomed in areas of the Patapsco River. So on the right, you'll see that there was significant chlorophyll all throughout the Patapsco coming out into the main bay, kind of headed north. The same pattern is shown in that RBD on the left, which is indicating that it is mostly a dinoflagellate bloom. And we were able to use satellites to help direct monitoring into those significantly more red areas. And it was a pericentrum minimum bloom. We're also working with NASA. There was an image that was supposed to show up before that, but the animation's not working. This particular summer, there has been a significant algae bloom kind of from the Patapsco all the way south to the Severn, hugging along the western shore of the Chesapeake. Sorry, that map's not showing. And so I worked with scientists at NOAA because it seems to be that it's been excessive and a large scale bloom. And over here, this is the year 2025 in this group, you can see that we've had a significant amount of area. So this is the satellite each day, how much area was greater than 30 micrograms per liter chlorophyll, which is a significant bloom. We've had a very extended period over that compared to past years. And so she was able to take that for that area of the bay and kind of work up a chart like this so that we can compare this. And eventually we're hoping to use this to look back in time for like climate changes of some of these chlorophyll blooms and duration. We're also, as I mentioned, working with a couple of imaging platforms. What's called the PhytoChap is an imaging flow cytobot located at the University of Maryland Horn Point Lab on the Chaptank River. There's the website if you want to go and see it. It is at a fixed site. It's not used for freshwater HABs, but it does suck water out of the Chaptank River once an hour and produces images like these. And then these images can be classified through an online portal called Ecotaxa, hosted by the French government, to help us assess if there's harmful algae bloom there. A very similar device was being used in Texas for red tides and accidentally detected a different species called Dinophysis that can produce diuretic shellfish poisoning at bloom concentrations, closing down that Texas fisheries just before a major festival was going to happen on oysters and hopefully therefore protecting human health. DNR is working with an imaging platform called the FlowCam 8400. It is designed for rapid algal identification and enumeration. This model is specific for cyanobacteria because it also uses fluorescence in both the chlorophyll A and phycocyanin wavelengths. There are multiple modes and magnifications, and so right now we are still working on designing what is the best process for this. And so to do that, you have to classify image libraries for each species and train the instrument to know what they are. And there's a whole bunch of statistics with each library. So these are a couple of example libraries from some of our freshwater work. This is an example of a microcystis colony, and they can be very variable, as you see. So training the model to understand and recognize these using some statistical parameters that are built into each measurement. So the next part I'm going to get into for the talk is talking to you more about some of the harmful algal blooms that we monitor for and have seen in the Maryland waters. So first, I want to talk about overall the harmful algal bloom management when we're in Maryland. So it consists of 3 different agencies. The Maryland Department of Health is connected to CDC and the One Health and Harmful Algal Bloom database. They also oversee the county health departments where actual authority for making any closures lies. You may be aware there's over 23 counties in Maryland, so we have to work with all of them when things happen. Then you have the Department of Natural Resources, where I sit. We conduct routine phytoplankton monitoring. We also get involved in harmful algal bloom responses. Most people think of us as the fish, the birds, and the bunnies agency. And then you have the Department of the Environment. They are the regulatory agency in the state. They also oversee the fish kills and have the toxin testing laboratory. They also coordinate with us and do algal bloom response monitoring. And then there's also sometimes a 4th agency. I put Maryland Department of the Agriculture in there because at certain times, depending on the harmful algal bloom, we do have to work in coordination with Maryland Department of Agriculture. There's also been times when there has been animal deaths, and again, that's in coordination with MDA. So what are some of the impacts of harmful algal blooms? There's human health, which can cause illness and irritation. There's impacts to animal, pets, livestock, and wildlife can die from these or become violently ill. And then there's also environmental impacts, including habitat, water quality, and recreational use. Virginia has actually declared some parts of the Shenandoah impaired due to algae blooms because recreational use was deterred. So as far as marine harmful algae go, I call them the dirty dozen, although I feel like the list continually is increasing, but I break them down into 3 different categories. So there's the potential toxin producers. There are ones that just produce high biomass, which can shade seagrasses, retain nutrients for regeneration, or lead to fish kills when the high biomass is decomposed, robbing the water of oxygen. There are also some that are just shellfish irritants. While they don't kill it, they significantly irritate the gills when they are filter feeding. And then I guess it's also under potential toxin producers, but there are certain ones that are fish killing. So they produce toxins that are only impacting fish, such as carlidinium, which was what was killing fish during Pfiesteria for those that were around back then. And the one thing I put the clam picture in to remind me to say is that often we are monitoring for these harmful algal because when shellfish ingest them, they can accumulate the toxins and then pass them on to wildlife or humans. So this is a table that shows the harmful algal bloom impacts. The algal species are first, but what's most important are these impacts that are outlined in red. The different toxins that they produce are in the 3rd column, but I think probably many of you have heard some of these impacts, including paralytic shellfish poisoning, neurotoxic shellfish poisoning, diuretic shellfish poisoning, amnesic shellfish poisoning. Hopefully nobody's had dinner, all this yummy talk. And then there's some that are just lead to fish and invertebrate mortality due to the ichthyotoxins that they produce. So all the way, the last column on the right is essentially showing what the cell threshold is that we use to then determine if we need to send samples for toxin testing. So with all of our routine monitoring, if any of these species exceed those cell thresholds, they'll then be moved on to further testing. Again, I'll reiterate that second to last column shows where the toxin level in the shellfish meat that would lead to a closure of the shellfish area. We have not had any in Maryland. So one thing I wanted to point out is that all of our data is uploaded into our database weekly and then can be found on these Eyes on the Bay harmful algal bloom maps. And so you can go in and change dates. You can filter to find certain species over time. But you can get maps like this that's going to show you the past couple of weeks where potential harmful algae was found. Now, again, if it's in orange, it's not exceeded a threshold. It has not produced any toxin. But if there is a red triangle, that usually means it is above those cell thresholds I mentioned, and we would be testing it for toxin. But you can click on any one of those points. So if you live up here near Harvard Grace, or if you live here on the Choptank and want to see what that was, you click on that button and you'll get a whole bunch of information of the sample date, the actual algae that was present, the current status, and what that cell concentration was. That can be compared. If you go to the legend, there's a way to see what all those thresholds are for each species. So you can compare that against. So this is a map, just a few of the maps that we've made that show the distribution of 3 species. If you go to the link that I showed you, there's a handout that shows the distribution for 12 of the common marine HAB species. But this shows you whenever these were exceeding those thresholds that I mentioned for each species. And as you can see, each of these species has a different threshold. And you can kind of get an idea of where they are found within the state. This map shows, the Prorocentrum here in the middle are the only ones that's shown in Virginia, but then there's some species that are only found in Virginia and not often in Maryland. And I'm going to transition to some of our freshwater species of concern. The freshwater are very interesting group. So many that can potentially produce toxin, and they really like to rename these ones a lot. But I'll talk more about the cyanobacteria, and then I'll have one slide on didymo or rock snot. For those that are more into the streams, might have already heard of that one. So freshwater toxins, they produce liver toxins and neurotoxins. And so this is a summary of the health effects of each of those toxins in humans. The most abundant algal toxin that we find in Maryland is microcystin. It's found throughout the state in many different areas. In the last couple of years, we've just started to find some of the neurotoxins, both anatoxin and saxitoxin, mostly within benthic mats. Most of these mats don't seem to be releasing those toxins into the water column, at least whenever we've had samples of both the mat and the water at the same time. But other states, including 25 other states, are also working on these benthic mats and these novel neurotoxins that had not been detected previously, but probably also wasn't monitored previously. But they have caused dog deaths in over 20 states. Again, you must ingest most of these toxins for them to have these health effects. So microcystin is typically found in those planktonic algae blooms. And so it's easier, especially for children or animals, to accidentally ingest those by ingesting some water while swimming, or animals will actually drink the water. These neurotoxins that are in the benthic mats are not always ingested either by shellfish or people. You'd really have to go out of your way to eat them, but some animals are attracted to them, including dogs. So as I mentioned, microcystin is the most abundant algal toxin that we find. The map on the right is summarizing the occurrence of microcystin over a 20-year period. The size of the dot indicates the number of years or times it was found in a particular location. The color is indicating the concentration of microcystin. So only the red dots were exceeding of the EPA threshold with the 8 parts per billion. The other thresholds are below that, but still interesting to show in terms of its total distribution throughout the state. It is important to remember that most of our water bodies are only monitored once a month, and algal blooms can change within a matter of 24 to 48 hours. And so our main motto is, when in doubt, stay out. We have some educational pamphlets and other things if you guys are interested in having as a handout in any of your events that kind of have pictures of what it looks like, how scums are formed, the coloration, streakiness, and things like that of the water. There is a paper that this is summarized in, and I can't see it on my screen because that black box is up, but hopefully if anybody's interested, I can send it out. It was done with Barnett Ratner at USGS. So one of the things that happens once we have a concentration that exceeds the EPA threshold is we work with the local county to issue a no contact advisory for that water body, and then continue sampling it until we have 2 water samples that are below that, and then it can be reopened. And so what I tried to do was to summarize we usually do 5 to 7 no contact advisories a year on average. So what I've done in this graph is try to summarize the number of days that we saw impaired use or a no contact advisory placed throughout the state. So the reason why there's more than 365 days is because there's multiple water bodies, and some might have been closed 30 days, some might have been closed 90 days. But it's just kind of trying to give an overall assessment of those number of impaired use days, kind of similar to what they do with beach closures and fecal coliform monitoring. And as you can see, it's highly variable from year to year, the number of days, and a lot of that has to do with the current conditions. So cyanobacteria tend to like it hot and dry. So if you get storms coming through that flush out the bacteria, or if you get cold nights like we've had this month, they'll keep the blooms at bay. So benthic mats is what's been keeping me busy the last 2 or 3 years. So I wanted to start with a little bit of images of what do they look like. Um, so they can be attached to rocks, but often we find them growing on the bottom. These are several different images of some planktothrix, some Lingbia or Microceria woolii in the Potomac River. On the bottom left are one of the most interesting ways it presents, and I call them Lingbia balls. The algae has come up early in the season somehow because of either tidal action or wind, that they form into these balls, and its name woolii is very appropriate. They're very woolly feeling, and they stay in that formation. I've put them in a mason jar, and they were still looking like balls almost a year later. The only other place I've heard them look like this in appearance was in one of the Great Lakes. So this is kind of what they look like. And the reason that we've been interested in them is that they've been shown to produce neurotoxins. So we've been studying this over the last 2 years in conjunction with partners at SUNY, University of Maryland, and other areas. And so what you're seeing here is a summary of all of our results. Since that's a whole separate talk that can take me 30 minutes. But we've been doing some specific lakes monitoring in the state. So most of the things to the left of the top box are from different lakes across the state and all over. And we've also done some sampling in the Chesapeake Bay, a lot of it in the Upper Bay and the Susquehanna Flats, but also the Potomac River. So these are just box and whiskers of the saxitoxin results found at each site. And what you can see there is around the 30 micrograms per gram line is a recreational contact threshold suggested by the World Health Organization for this. Again, you would need to ingest it, and most people aren't taking those mats and eating them. But it is something of concern, and we're not really sure what the downstream impacts are or the environmental impacts are. I forgot to mention when talking about microcystin that it's been shown in macroinvertebrates in the stream, as well as birds that were eating the macroinvertebrates along the stream in Virginia. So saxitoxin we did put in to try to look at it to see if it's moving, and we weren't funded because there's not a lot of harmful algal bloom funding right now. But it is of concern if this were to move or if it were to accumulate in shellfish. Saxitoxin, with several of these algal toxins, there can be up to 100 variations of the same toxin with different potencies. There is also marine saxitoxin that's produced by Alexandrium. And when that accumulates in shellfish, it can cause paralytic shellfish poisoning. We don't think that these filaments are getting ingested by clams or other shellfish because they're just too large for them to filter, but they could be getting into other wildlife. We did do some testing of amphipods that were growing in the Microceria mats up in the Susquehanna Flats, and they were positive for saxitoxin. However, they seem to be functioning okay. So more work to be done. The other neurotoxin is, what I forgot to say about saxitoxin is that it is more potent than cyanide. Anatoxin is the other neurotoxin produced by cyanobacteria, and its nickname is very fast death factor because dogs and other animals, when they ingest it, die within hours or minutes, where microcystin might take longer. Again, the different water bodies are along the bottom, and the toxin concentration is on your y-axis. The World Health recreational contact threshold is that red dotted line at 60. The orange bars is anatoxin that was detected within benthic mats, but the darker brownish red is samples that we actually tested in the water column. The water column anatoxin were due to different species, not benthic species. It wasn't in conjunction with the same mat samples, there was a different species present in the water that indicated to get it tested. The thing that is interesting is that California set a lower threshold, as you see at the very bottom there in yellow, that they called their dog threshold. So as I mentioned earlier, antitoxin has been the factor in dog deaths in about 20 states. None in Maryland, but we have had some dog death due to microcystin historically. The antitoxin was detected at significant concentrations in the Potomac River, both in 2003 and 2004, leading to an animal safety alert being issued along the Potomac. And this was what was posted about that and sent to different municipalities along the river. It is difficult because these they can vary over different stretches of the river in terms of their percent cover and in their toxicity. Even within the same mat or bloom, you can have toxic and non-toxic cells. So it's never a given that there's toxin given a certain concentration. So just to summarize, some of the impacts that we have seen from these cyanobacteria and their toxins is we have had dogs kill in both Dorchester County and the Sassafras River, and previous to the ability to test for these toxins, there were some suspect cases both on the elk and the sassafras also. In 2012 and 2023, microcystin killed birds and muskrats on Poplar Island. In 2004, again, before we were doing some of the animal testing, there were unconfirmed deaths. On average, I was saying 7, but it's been more like 5 to 7 per year. No contact advisories on different water bodies. Some of our reservoirs have also had advisories, so drinking water sources. Fortunately, most have not been being used for drinking water at the time of impact, but we work very closely with the, whoever's in charge of the drinking water for that area. I talked about the cyanotoxins found in the benthic mats and the microcystins that have been found into the food web in the James River and in the West Virginia portion of Potomac. So one freshwater harmful algal that I couldn't do the presentation without mentioning, even though we don't routinely monitor for it anymore, is didymo, which is actually a stalked diatom. And its lovely nickname is rock snot, but you can kind of see by looking at the rock being held why it got that name. So in 2014, when this was found in Maryland, we used eDNA to help track if it was spreading from the original source. We did find it in some other locations. These are just all of the monitoring sites that are in this, both by Frostburg in purple and DNR in green. But it did lead to a ban on felt sold waders, and it established the different wash stations at entry points to certain streams that you may have seen around the state. This is the algae that was responsible for those. So I've said a lot about all these algae and harmful algae and phytoplankton, but as Natural History Society people, you might be saying, wow, well, when it comes to phytoplankton ecology, is there any order, or how can we deal with all of this? And so one of the really interesting conceptual ideas is called a mandala. And there's a very well-known mandala by Margolef that kind of showed that both nutrients and turbulence kind of helped to determine what was happening, but this was much more oceanic, but it was also just trying to see why did red tide become dominant at times. And so if you look at the turbulence along this, nutrients along this, you change from from the diatoms over to the rounded dinoflagellates to the flattened dinoflagellates. But without getting really into depth in that, Dr. Patricia Gilbert down at University of Maryland Horn Point Lab is taking this very oceanic kind of mandala and increase that to estuarine phytoplankton, which if you've never heard before, the nutrient ratio has a lot to do with phytoplankton ecology. So very much similar to when you're doing your home garden and your tomatoes need a certain ratio of nitrogen to phosphorus that might be different than your herb garden or your flowers. Phytoplankton are very much the same, and the ratio of nitrogen to phosphorus can actually change which phytoplankton are dominant. Redfield was the one who came up with this ratio, and the most balanced is a 16-to-1. But as Dr. Gilbert showed, as you change the dissolved nutrient ratio, the N to P ratio, you might change your phytoplankton. But she also looked at organic and inorganic nitrogen sources, and that ratio also impacting what algae populations win the competition, right? Because everything's a competition in nature for resources. So over on the right, you have more of these diatoms that need more mixing to stay in the water column and not as much nitrogen. And then over here, you have some of those picocyanos, but she expanded that. Yeah, she went from that one to this. So in addition to that dissolved nutrient, she had light adaptation, their mobility, she has temperature ratios and other pigments. But all of this is trying to just put into context a conceptualized way of understanding which phytoplankton and what factors may influence what phytoplankton are the ones that bloom. And then just to take it one step further out and zoom out even more from the microscopic, she started adding in what different zooplankton then might graze on the different phytoplankton that are at the source of this food web. And so to the left, you'll see that it's more reduced nutrients, regenerated nutrients, which led to smaller, maybe copepods and other mesozooplankton. But if you had the right conditions that led to the diatoms and you had more new nitrogen entering the system, you might get bigger copepods, new production that is better for fisheries production. It makes bigger fish. So that's how I'm going to just end that, you know, that mandala is just kind of the pattern, kind of symbolically representing our phytoplankton universe that Dr. Gilbert has kind of synthesized. I know that was a lot in a short time. I am happy to answer any questions.
Well, thank you so much, Cathy. You can unshare, come back. And we have questions in the chat box already, and we'll let me go back and okay, let me put a spotlight on you. So everybody, and again, if you want to ask a question directly, just raise your hand and I'll call on you, or you can put it in the chat box. But that was fascinating and kind of scary, but fascinating.
I tried to say at the beginning, we haven't had any. So other than the water contact advisories, which we do to try to prevent any health impacts.
Yeah. I like that.
But I think everybody walks away from my talks with protect your dogs and kids from any scummy water.
Jason asks about the fuel, the food for the algae. Where is it coming from to get these blooms? Is it urban areas? Is it agriculture areas? Is it something else? And how is that being used to control the harmful algal bloom?
Yeah, so it's definitely specific to the different algae in different areas. So of course, I will say the nutrients are coming from all of the places, but we do have a total maximum daily load for the Chesapeake Bay that's been developed, or the nutrient diet that looks at all of the sources of nutrients and tries to reduce them to keep that chlorophyll A level at the right level. So that's one answer. But the more we do in our lakes, which isn't routine monitoring, but has been more specific targeted monitoring, is that a lot of these lakes have filled in over time. They were made with dams that have filled in for 50 to 100 years now. There is a lot of sediment in those dams that are regenerating nutrients. So a lot of the blooms are being fueled also by those regenerative nutrients from the benthic or the sediment that's accumulated, and it's high in phosphorus. So when these blooms make an anoxic layer in that lake, all of those nutrients start pumping out. Or as Jeff Cornwell's shown on the sassafras, when pH exceeds 9, the same phosphorus is released from those benthic mats. The other thing with some of the benthic mats that is a hypothesis of mine, so don't quote me or put me in a paper for this, but we've cleaned up the bay and decreased water column algae. We've shown that in trends. So we've decreased some nutrients, but we did target nitrogen, right? So most of our goals have been nitrogen focused. They've just recently added phosphorus and are starting to work more on those. But as I mentioned, that red field ratio. So as you decline that or you increase the nutrient, the N to P, because you have a lot more P than N, there's certain cyanobacteria that can fix their own nitrogen, and Microceria is one of those. So up on the Susquehanna Flats, one of our hypotheses is we decrease the water column nutrients, which let more light grow at the bottom. And then when there's a nutrient limitation, the cyanobacteria can outcompete the others. They also make an anoxic layer right at the sediment water interface, which allows the phosphorus to regenerate, but it doesn't get into the water column for other algae. And so that Lyngbia kind of beats the other algae in that particular situation. So that's a long answer without a direct answer. I'm not a politician, I swear.
Can I ask, I mean, if as the reservoirs fill up, and that's decreasing the amount of water storage capacity for the reservoirs, that process is going to continue. And are they dredging the reservoirs? And if so, does that throw up all the other things into the water and all that kind of stuff?
Most do not have a dredge plan. There have been some that have been dredged, and usually it's been taken off site, so it hasn't really stirred anything up from the bottom per se. But that is something, even with our state-owned properties and our parks and stuff that you go to, we need to come up with a plan on, are they going to naturally fill in, or are we going to dredge them to contain or continue to use them as a lake?
Okay. Now Jason asks about their eyes on the bay data. Is the general public allowed to download the data in the form of a CSV file for further GIS analysis?
I don't think the HAB data can be downloaded as CSV, but I'll have to check with our web person to make sure. They were trying to get that, but I don't know if it actually happened. I don't remember seeing that it was visible to do that.
And then we had a question about that. I mean, it was pretty amazing. You said there was over 1,500 different types of algae. And have you seen all 1,500 types yourself?
Yeah, so I am not our resident taxonomist. I do look at samples, but we have somebody who's been with us a few years, but that number was developed from Harold Marshall with Old Dominion University, who looked at both Virginia and Maryland long-term phytoplankton data in our databases.
Well, are there any specific differences in the saltwater versus freshwater versus estuarine algae differences or similarities in them?
I mean, there's definitely differences. I talked like we have a whole different group of freshwater species. That's not included in that 1,500. I think Harold Marshall had done some freshwater work, but most of that is the estuarine species. I'm not sure what other differences. I talked about their ability for the nitrogen fixation by some of the freshwater species. That isn't something that occurs in the salt. Some of the estuarine species, I didn't go into this, can be mixotrophs. So they can photosynthesize when those conditions are right, or they can be feed me, see more, and start eating other stuff when they need to. And so that's actually been a very interesting group, mostly dinoflagellates, that can do that. And if those, remember, Pfisteria also could do that.
Wow, that's cool. And then let's see, Christina asks, is this one of the reasons why we see recalls on food supply, like shrimp or crab at Walmart?
Often it is not due to algal toxins. There might be like fresh harvested clams up in Maine, sometimes have saxitoxin, but they are so good at monitoring, those things never hit the shelves.
Okay. Jerry asks, why is the NIH threshold for antitoxins so much lower than the World Health Organization?
I think it wasn't a formal one, but it was one that was put out in a paper as probably more cautionary. But it's not a nationally adopted. It was just in a paper, so I probably shouldn't refer to it that way.
All right, Jason asked, can you do a little refresher on K and R succession graphs again, please?
So R usually make lots of babies, right? And that's how they want to propagate, and K will nurture and take longer in that. That's my quick summary of my ecology.
Yeah. Jerry also follows up with, do I understand the mandala correctly in that if the NP is balanced or more N than P, and the nitrogen preference is strongly more NO3 or inorganic, then we should have fewer phytoplankton?
Not necessarily fewer, because concentration of those nutrients matter, but it'll probably swing more towards diatoms, but you still need turbulence. If there's not turbulence to keep the diatoms, because they are glass houses that like to sink, where some of the other species either have gas vacuoles to help regulate them in the water column or flagella.
And it was fascinating to see all of the new technologies, and especially with the AI training the models to look at that. Jean was saying that the monitoring has changed dramatically since he was collecting water samples 40 years ago. And I assume that it also allows these new technologies to do more extensive and monitoring and better, not better monitoring, but more prolific monitoring than just having a person out there going and checking individual stations.
We're hoping. We're still in this process of training and getting to that. But that is our goal, is to kind of bring things into the 21st century and use some of these new technologies to help us, because we got to a point where we were doing almost 1,200 samples a month, and we only have one taxonomist that does it full time. So we had to cut back some of that sampling. It was not maintainable long term for somebody's sanity, and there was no funding for another taxonomist. And so we're hoping some of these technologies will help us quicker, more rapidly process the samples and make sure we are on top of things.
Steve asks, what's being done to encourage anglers and others to clean their watercraft, boots, and other equipment after each use to prevent the spread of aquatic invasives?
Yeah, so we had an extensive program at Deep Creek, more related to water chestnut, which is an invasive submerged aquatic vegetation that can get hold in certain water bodies. And then our SAV group was also physically going to ones that were infested and removing it to try to decrease that. Also, I mentioned that they put out those waiter stations where you're supposed to clean your boots when you go from place to place, trying to reduce the spread of that didymo, which did wind up going to several other areas, unfortunately, probably because there is no enforcement really of people to clean the boat for that. There was a couple of check-ins at Deep Creek where they were looking for those invasive plants, but it's very difficult to know if you moved a harmful algae from one place to another. And honestly, other people will say that the birds alone, in terms of cyanobacteria and moving them, I didn't even talk about golden algae, which has been detected with eDNA in the state, kills massive fish kills in aquaculture ponds down in Texas and more southern states. we know it's here, but again, we haven't seen conditions that maybe make it bloom.
Jean also says that a while ago, the Baltimore looked at potentially dredging Loch Raven Reservoir years ago, but it wasn't cost effective. And he asks, did Didymo eventually get into the gunpowder falls between Pretty Boy and Loch Raven?
Yeah, so 2 points. Yeah, the cost effectiveness has been very difficult for dredging, even like convincing the state to dredge. We've done some very small dredge projects in our state-owned lakes, but I do know Columbia Association is one of the only ones that actually went in and dredged. But essentially, their ponds are Lake Elkhorn, like they're really stormwater management ponds. And then I do believe Didymo did get into gunpowder, but I would have to go back and double check my Didymo results to make sure. It did spread to several other areas, and I think even we found it in Savage one time.
And it's so fun to say Didymo. That's a fun word.
It is actually fun to say.
And Alan has his hand up. Alan, what you got for us?
Hi, Cathy. Thanks so much. And 2 questions. One, I'm not sure if I'm going to be using the right term, but I was visiting Sarasota several years back, and there was some kind of a red tide. And I know that I immediately at the beach, I was started coughing, and then within half a day, had a horrible nodule in my throat. And I'm wondering if that could have come from that, number one. I know you're not a doctor and, all of that. And then the second question is a personal question for you, just how you got into this field, your interest, where did your interest grow from? Not unlike algae growing, okay.
And so the first one, Sarasota, gets Karenia brevis blooms, which produces a toxin called brevetoxin, which absolutely is a respiratory irritant, and they put beach signs up. So their red tide is not the same as when people see red tides here, and that's why I don't like color names, because many things can produce the same colorish water. So yes, your coughing was most likely related to brevetoxin being aerosolized at the beach due to wave interactions. Fortunately, not a lot of our toxins do get aerosolized, but brevetoxin is the one that most people look to in terms of that information. In terms of where I got interested, definitely was always interested in estuarine work. I actually went to Salisbury University, having lived down in Virginia, and then I moved there. Then I did my graduate work at University of Maryland on ecosystem metabolism. So looking at oxygen and carbon dioxide changes from primary productivity. Started my career at DNR overseeing coastal water quality monitoring and brown tides, which I didn't even mention today, caused by a very small organism called Oreococcus, impact shellfish because it's too small for them to eat. And so working on that down in the coastal bays just kind of elevated my interest and also my job pathway at Maryland DNR.
Jason is a recent graduate from UMBC looking for early career jobs and wondering whether I can get in wondering whether I can get in contact with regarding to this.
Yeah, sure. Happy to talk to new graduates. I know it's a terrible time probably to be getting graduated and looking for jobs. One of the things we've used in our lab the last 4 years is called the Chesapeake Conservation Corps through the Chesapeake Bay Trust, which is great for new graduates. It aligns you with a host organization where you get a full year of experience, which helps build your resume, help getting future jobs. So yeah, definitely.
Jerry asked, are there any phone apps that can reliably tell if an algae mat is dangerous or not?
No, not to my knowledge. We have a couple of field guides, and actually George Mason and ICPRB are looking to update those, and I'm hoping at their upcoming conference this October, they'll have a final field guide because some of our stream people also would like that. But as I also mentioned is, even if I know that it's a particular cyanobacteria that's a potential toxin producer, I don't know if it's producing the toxin by looking at it. Again, all of these blooms can be 100%, almost never 100% of the cells producing toxin, or they can be 50-50, or they're still very curious what turns on and off that gene to produce the toxin, because as you can imagine, that's a very high energy use for a cell to make this. So it has to have an ecological benefit. Most people assume it has to do with avoiding predation, and that. And so Judy O'Neill over at Horn Point University of Maryland hypothesizes with the Lingbia, it puts more energy into that early in the season, making more toxin like in June for the benthic mats. And then as the season goes on and it has a lot more established, it doesn't produce as much toxin, but we're still trying to figure it out.
And where can we go for this, the citizen monitoring, the hydrocolor thing, if we wanted to get involved with that? Is that open to anybody, or do we have to take a training for it?
Yeah, no, the hydrocolor app is just a NASA app. There's a bunch of people with CERC volunteer monitoring using it, trying to help just calibrate the NASA models because of all that atmospheric correction. So I think we bought the gray cards, the photographer gray cards on Amazon for like less than $10 and downloaded the free app called Hydrocolor. I think it has a water. I'm trying to look on my phone to see if I can see it. It looks like a water droplet with a black background and a rainbow color. I don't know if you can see it. And it has, doesn't really show up too good. And then, yeah, we've done some demonstrations where we train people on how to use that. So if that's of interest, me or my staff could come out and show that. I'm hoping eventually that might help us, like with those different bandwidths, tell that we have a dinoflagellate or something in that red band difference that's showing a potentially harmful mat, or something that's showing that fluorescent green cyanobacteria. I'm hoping that the data might eventually get used into an app like that, but currently it doesn't.
And along those lines, and what Jerry was saying, are these the macro algae, are they are people posting them on iNaturalist or anything like that? Or is that a useful tool for you or for people in terms of learning and seeing and recording what they see?
There is actually another app called Cyano app that reports things that started up in the New England area, but it's expanded. And so if somebody reports to that app, I'm actually the contact and will get notified, and then I may or may not follow up. I have gone on iNaturalist and found some people's observations, which were very useful. But it's not like I get notified or I know they're there. And so reporting them in both places is probably good for documentation.
Christina asks, are there any links to cancer with the algal blooms and exposure to the toxin?
Yeah, so there's especially with cyanotoxins and microcystin in particular, it is a known tumor producer and potential cancer in mice and rats, but there's not been a lot of long-term studies on some of these to know for sure, but that one I know for sure has been shown.
Would you mind putting your email in the chat box? There's an ask for that. That's a possibility. All right. Any other questions on algae or anything tonight?
Yep. So like I showed, the Eyes on the Bay website also has an online portal that if you do have a bloom, you can report it online, which we think people, and you can upload pictures to that or email them to us, and then we'll be in contact to either do that. If it's a fish kill or something you think is really harmful, there's 24/7 health hotline, a phone number that you can call, and somebody will respond.
What is the name again of the cyanobacteria app you just mentioned?
Let me double check I give you the right one.
That's from Jerry.
Thought it was cyan, but I'm finding cyano. Yeah, it might be Cyano Lakes, but I feel like that's not the one. I'll have to get back to you on that. I'm not finding it on my phone real quick.
Oh, okay. hopefully I'll let me know, and I can pass it along to folks.
Okay, sorry about that.
That's okay. We're all into helping be out there. We're out there observing and recording things. So anything that we can do as community scientists to help collect data and push the understanding forward, we're all about that.
That is awesome. We can always use those extra eyes on the bay. We work a lot with river keepers and county professionals for the same reason. They see those areas that they're familiar with more often.
He asked, is it cyano alert?
I'm not finding it. I don't know.
Okay.
Sorry.
That's okay. We'll find it and we'll let you know, folks. But thanks. Any other questions? All right. Well, you have
Thank you so much.
certainly it made us all a lot smarter, and we're going to look at water differently and breathing differently and all kinds of things. So thank you so much for sharing your time and expertise with us tonight. Thanks everybody for coming in for our new season of Must Learn Thursdays. Next week, it's the Appalachian Cottontail. So come back and we'll learn all about that. Take care,
Thank you.
everybody.
That was a great talk.
Thank you. Thanks so much, everyone.